Okay, so a bit of a change of tack now from what we've been talking about so far. Rather than looking at epigenetics and how these marks are inherited over time, this is over a much smaller time scale. So now we're looking over changes during the day that aren't necessarily inherited between generations. Okay, so before I got started, I thought I'd talk more about why the circadian clock is important. So work done here in Cambridge by Alex Webb and Anthony Dodd has shown that if you have a circadian clock that's in sync with your environment, you're actually a lot fitter than you would be otherwise. So circadian mutants that are held in a different environmental setting to 24 hours are actually less able to set seed and enhance their biomass over time. The circadian clock is actually pretty pervasive, and it affects approximately 1/3 of the genome, and it affects a broad suite of different mechanisms at both a physiological and gene expression level. So here we're looking at a bean plant that's been grown in light-dark cycles for 6 days and then transferred into constant light. And you can see that even in the absence of these environmental cues, it's continuing to put its leaves out, and then they drop down during the subjective dark. And we similarly see circadian control of photosynthesis and also plant growth. Okay, so as well as looking at it at a phenotypic level, we can also look at a gene expression level. And we do this routinely in our lab using luciferase reporters. So on the left-hand side, you can see a luciferase reporter gene under the control of a circadian promoter. And we can monitor the amount of light given off by that plant over time. So we're looking over 6 or 7 days here. And you can see that we get a peak of expression during the subjective evening. So for this first day, we're in a light-dark cycle, and then we've been put into constant red light, and we can measure a very nice oscillation over time in these plants. So from this waveform, we can generate a lot of different data, but what I'm going to be talking about mostly today is the period length, which is simply the time it takes for the circadian clock to go through a single cycle. And because this oscillation continues without environmental inputs, the differences in period length give us a lot of information about how the internal clock is affected in different mutant backgrounds. Okay, so the circadian clock has evolved many different times over evolution in many different lineages, but in each case you have a common organization. So you have interlocking negative feedback loops that occur both at a transcriptional and a post-translational level. So how this works basically is you accumulate protein A, which represses transcription of gene B in this case, and then over time this causes A to stop being expressed and is gradually degraded over time. Once A has been degraded, you then promote expression of B again, and then you get antiphasic expression of these two genes, which gives you a 24-hour rhythm. Okay, so it isn't quite that simple in plants. Of course, there's functional redundancy built into this system. And so this was the first clock that was identified in plants almost 10 years ago now in Steve Kay's lab. What he found was that there were two MIB-like transcription factors called CCA1 and LHY that bind directly to the promoter of TOC1, which is expressed in the evening. And then TOC1, through some sort of indirect mechanism that we still don't fully understand, promotes the expression of CCA1 and LHY the following morning. So although this original loop has been identified for several years, there are still unknown components. Recently, it's been shown that ELF3 is involved in the evening part of the clock, as is ELF4. So there are posters here about that, I noticed. And there are also morning components as well that I won't go into talking about today. Okay, so why was our lab interested in studying Jumanji? Well, Jumanji was originally identified from a bioinformatics screen using publicly available microarray data to find genes that were co-expressed with known clock components. So we're looking here over diurnal time in constant light, and then we're looking at the expression of these three genes over circadian time. And you can see that TOC1 in the green ELF3 in blue, and also Jumanji are all expressed in the evening phase, and they all have a pretty nice circadian rhythm. ELF3 is a bit less rhythmic in this data set, but by qPCR that looks better. The most interesting thing about looking at JMJD5 and TOC1 expression is that not only are they correlated over diurnal time, but they're also correlated over developmental time. So here along the x-axis, we're looking at TOC1 expression, and up on the y-axis, we're looking at JMJD5. And whenever you have high levels of TOC1, you have high levels of JMJD5. And these are from over 50 different tissue samples, so these are from roots, saliques, leaves, all the way through development of Arabidopsis. So it's kind of guilt by association. Jumanji is co-expressed with TOC1, and TOC1 is known to be involved in the clock. So we went ahead and identified mutant alleles within the Jumanji gene and looked to see whether we saw a circadian phenotype. And indeed we did. So in wild type, in constant conditions, these plants are oscillating with a period of about 25 hours, which is close enough to the 24 hours that you would see in entrained conditions. But in our two mutants, what we see is we see a significant reduction of about an hour and a half in constant red light. So it would appear that Jumanji is also involved in the circadian clock, although we're still not quite sure the mechanism by which this is occurring. Interestingly, we also saw that this was dependent on the fluence of red light we were growing our plants under. So this is a fluence rate response curve, and so what we're doing is looking at the period length of our plants under gradually increasing levels of red light, up to about 50 microeinsteins at this far end. And so what you can see under dim light and actually in the dark, we don't see any circadian phenotype at all. Whereas as you increase the fluence levels, we get a greater and greater difference between our wild type and our mutant. So there appears to be an environmental factor as well into the phenotype that's controlled by Jumanji. And in fact, when we turn the lights as high up as we can go, sorry, this has been turned off, this is at 120 microeinsteins monochromatic red light, which is pretty artificial. But it does mean that we completely lose rhythms in our Jumanji plants. And you see the same thing in TOC1 mutants as well. By contrast, wild type is still oscillating under these conditions. So it looks like our Jumanji mutants are more sensitive to red light. Okay, so what I've told you so far is that Jumanji is co-expressed with central clock genes and that it also has a circadian phenotype. And interestingly, this appears to be an interaction with environmental factors as well. So under very high levels of monochromatic red, we're seeing arrhythmia in our Jumanji mutants. Okay, so what do we think Jumanji is actually doing biochemically? Well, JMJD5 has a conserved Jumanji domain, and these have been associated with the demethylation of various histone marks. So what I'm showing here is a phylogenetic tree of JMJD5 orthologs in different species. And the interesting thing is that they all cluster together regardless of which species they're from. And they tend to be a single copy in each of these different genomes. So Arabidopsis, rice, and poplar JMJD5 cluster together. In this analysis, because I've cut down on the number of Jumanji orthologs I've included, the Drosophila Jumanji is actually closer to plant, although you don't see this in a bigger tree. And then you have frog, mouse, and human JMJD5 as well. And these are all closer related to each other than they are to the closest ortholog, which doesn't contain the residues necessary for the histone demethylase activity. And the conservation is so close that we can overlap the human predicted structure in red and the plant structure in blue, and they overlap pretty well. And I'm also showing that all 5 of the residues required for histone demethylation activity are actually conserved between the plant and the human forms. So what a group based in Sacramento did last year was that they were able to show that the human form of JMJD5 is a histone demethylase. Specifically, it demethylates lysine 36 of histone 3, and it takes off a dimethyl mark and produces the monomethyl version. So how they did this was they ran this through a mass spec, either before or after incubation with the human version of Jumanji. And so this is beforehand, and you can see that you just get a single cluster of peaks corresponding to the peptide with the lysine dimethyl mark. When you add in JMJD5 to this mixture before you run it through the mass spec, you see that you get a new peak arising, which is about a 15 Dalton decrease, which corresponds to the loss of a methyl group from the peptide that was put into the machine. Okay, and what we were able to show with some collaborators down at the Salk Institute was that actually the human form of Jumanji also has a circadian phenotype. So these are human cancer cells that have been synchronized with a serum shock and then transferred it into constant conditions. And if you compare this peak here with the final peak on day 4, there's a greater difference in time when this peak is occurring, suggesting that there's actually a period defect in these Jumanji lines that have been knocked down, that have been knocked down for the endogenous JMJD5 compared to wild type. So what we had was a mutant phenotype that was consistent in both cases, and we were curious to see whether the enzyme activity was sufficient to rescue the phenotype either way around. So what we did was I put the plant form of the gene into these cancer cells and then knocked down the endogenous human form and to try and see what phenotype we could see. So on the left-hand side, we have either wild-type sarcoma cells or sarcoma cells expressing the plant gene, and these have been transfected with a scrambled siRNA as a mock control. As I've just shown you, if you knock down the wild-type cells with a JMJD5 siRNA, you get a shortening of period of about an hour. But interestingly, if you do this with cells that are also expressing the plant version of the gene, these are much more resistant to the knockdown, and the circadian period here is much greater than in the control. So this would seem to suggest that at least partially you're able to rescue the circadian phenotype by adding in back in what is presumably an active enzyme. We can also do this the other way around. So here we have wild type, so we're back in plants now. We have wild type plants and Jumanji mutant plants with an hour and a half difference in circadian period. Reassuringly, if we put the plant version back into plants, then we rescue the phenotype. But interestingly, we also see a similar effect when we put the human form of the enzyme back into plants. So it's not fully rescuing the phenotype, but it is significantly longer than the mutant controls. So this is pretty interesting. This suggests that enzyme activity, at least in part, is responsible for the phenotype we're seeing. Okay, so what I've told you is that we think Jumanji is a component of both the Arabidopsis and human circadian systems, although we're not actually sure where within the human clock Jumanji is acting. Each ortholog, when expressed in the reciprocal system, seems to restore activity, at least partially. And this is most likely because JMJD5 is a histone demethylase that's altering circadian loci. Okay, so I'd like to finish by thanking Stacey, whose lab I work in. Julian is another PI at UC Davis who helped a lot with the stats work associated with this. And Mike Covington started this work before he left Stacey's lab. And Sachin did the work in the mammalian systems at the Salk Institute. Just finally, I got a travel grant to come here, so thank you to UC Davis, and work in Stacey's lab is funded by the NIH.